Imagine a world where diseases are edited out of existence, crops grow bigger and stronger, and the very building blocks of life are within our control. This isn't science fiction. This is the brave new world of CRISPR. For years, scientists have dreamt of editing genes with precision. Now, thanks to a revolutionary tool found hiding in bacteria, that dream is becoming a reality. CRISPR isn't some clunky machine or vat of bubbling chemicals. It's a clever trick bacteria use to fight off viruses, and we've turned it into genetic editing superpowers. Are you ready to dive into the future? Buckle up, because we're about to explore the incredible world of CRISPR, the tool that's rewriting the code of life. In the annals of medical science, few discoveries have sparked as much excitement, potential, and controversy as CRISPR. Our journey takes us back to the early 20th century, to the birth of bacteriophage technology. These viruses, which prey exclusively on bacteria, became the unsung heroes of genetic research. By studying their interactions with bacteria, scientists unlocked the secrets of DNA manipulation. This was the genesis of genetic engineering. As the decades rolled by, this nascent field began to mature, setting the stage for a series of groundbreaking breakthroughs. Among these, the advent of recombinant DNA technology in the 70s proved pivotal. This technique allowed scientists to splice together DNA sequences from different organisms, creating a whole new world of possibilities. It revolutionized pharmaceutical manufacturing, enabling us to produce life-saving drugs like insulin and growth hormone in large quantities. However, these techniques, as innovative as they were, had their limitations. They were often imprecise, laborious, and time-consuming. The world of genetic engineering yearned for a tool that was precise, easy to use, and versatile. And in the early 21st century, that tool arrived in the form of CRISPR. CRISPR, short for clustered regularly interspaced, short palindromic repeats, is a system borrowed from bacteria. It's their defense mechanism against bacteriophages. But for us, it's a molecular scalpel, capable of making precise cuts in the DNA sequence. It's like a text editor for the genome, allowing us to add, remove, or change specific parts of the DNA sequence. This revolutionary tool has transformed the landscape of genetic engineering. It has made gene editing faster, cheaper, and more precise than ever before. It has opened up a new universe of possibilities, from curing genetic diseases to enhancing agricultural productivity and creating biofuels. But it has also raised a host of ethical and regulatory concerns, forcing us to grapple with the profound implications of our newfound power. The dawn of CRISPR has opened a new chapter in genetic engineering, a chapter filled with immense potential and profound questions. CRISPR, in its elegant simplicity, is a masterstroke of nature's engineering. This revolutionary technique allows for precision editing in the genetic code of life, akin to a molecular scalpel. Our journey through the mechanics of CRISPR begins with Guide RNA Design. Crafted in the lab, the Guide RNA is a custom-made courier, its sequence of nucleotides designed to match a specific location within the vast labyrinth of an organism's DNA. Its mission? To guide the Cas9 protein to the correct address within the genome. Next enters the Cas9 Protein Delivery. Cas9, the molecular scissors of the CRISPR system, is introduced into the cell. This enzyme is a marvel of nature, capable of being transported across the cell membrane and navigating the crowded cellular environment to rendezvous with the guide RNA. The magic unfolds with target DNA recognition. The guide RNA, bound to the Cas9 protein, scans the DNA, searching for its match. When it finds the complementary DNA sequence, it binds, holding the DNA in place. The climax of this intricate dance is the DNA cleavage. The Cas9 protein, now firmly anchored, makes a precise cut in the DNA strand. This cut is not random but meticulously calculated, aimed at a specific gene or genetic sequence. But nature abhors a break, especially in something as vital as DNA. This leads us to the final act, DNA repair. The cell, in its infinite wisdom, recognizes the breach and rushes to mend it. However, during this repair process, scientists can introduce a new genetic sequence, effectively rewriting the genetic code. This is the elegance of CRISPR, a system that harnesses the cell's own machinery to enact change. Each step, from guide RNA design to DNA repair, is a testament to the ingenuity of scientific exploration and nature's own engineering prowess. Thus, CRISPR stands at the frontier of genetic manipulation, a powerful tool in the hands of science. The applications of CRISPR extend far beyond the confines of a laboratory. Its potential is truly staggering. Consider gene therapy, where CRISPR offers hope for treating, perhaps even curing, once intractable genetic diseases. Imagine a world free from the chains of cystic fibrosis, muscular dystrophy, or sickle cell anemia. But the promise of CRISPR doesn't end at our own genome. Its influence extends to the very food on our plates. By precisely editing the genes of crops, we can create plants that are more nutritious, more resistant to pests, and better able to withstand the rigors of climate change. And let's not forget biofuels. By tweaking the genes of algae and other organisms, we can produce cleaner, more efficient fuels that could help combat the looming threat of global warming. Yet, with such monumental power comes the potential for misuse. The prospect of home-based genetic tweaking is tantalizing, but it underscores the urgent need for thorough regulation and oversight. CRISPR, thus, holds the promise to reshape our world, one gene at a time. With great power comes great responsibility, and CRISPR is no exception. In the age of gene editing, the ethical and regulatory concerns are as vast and complex as the technology itself. The potential for misuse of CRISPR technology is a reality we cannot ignore. Imagine a world where genetic modifications are not limited to curing diseases, but extend to designing our offspring with desired traits, a practice known as germline editing. The dystopian idea of designer babies is no longer confined to the realms of science fiction. Moreover, this technology could be exploited for malicious purposes. The potential for creating dangerous organisms or bioweapons is a chilling prospect that underscores the urgent need for stringent oversight. Yet the very power of CRISPR that can be misused also holds the promise to eradicate genetic diseases, enhance crop yield, and address myriad environmental issues. So, how do we balance the potential benefits with the potential risks? This conundrum has sparked intense debates within the scientific community. Some advocate for a moratorium on certain uses of the technology, while others argue for the need of a robust ethical framework that evolves with the technology. The stakes are high, and the decisions we make today will reverberate through generations to come. The question is not whether we should use CRISPR, but how we should use it responsibly. The regulatory landscape is evolving, albeit slowly. In many countries, laws and guidelines are being formulated to govern the use of CRISPR. However, these regulations vary greatly across borders and are often outpaced by the rapid advancements in the field. This underscores the need for international cooperation. A globally coordinated effort is required to establish universal ethical norms and regulatory standards. As we stand on the precipice of a genetic revolution, the way we wield the power of CRISPR will shape not just our future, but the future of life itself. We must tread this path with caution, wisdom, and a deep sense of our shared responsibility. CRISPR's potential goes far beyond fixing diseases. Here are some mind-blowing examples. Scientists used CRISPR to insert genes for bioluminescence, glowing, from jellyfish into pigs and plants. Imagine glowing houseplants or medical imaging with bioluminescent cells. Researchers are using CRISPR to edit genes in mosquitoes that make them resistant to the parasite that causes malaria. This could be a game changer in the fight against this deadly disease. Scientists are exploring using CRISPR to insert mammoth genes into Asian elephant cells to create a cold-resistant hybrid, a controversial attempt at de-extinction. CRISPR is being used to develop crops that are drought-resistant, more nutritious, or even produce biofuels. Imagine super rice needing less water or corn with built-in vitamin A. Scientists are using CRISPR to engineer bacteria that can kill antibiotic-resistant superbugs, a potential solution to a growing global health threat. Whoa, that was a mind trip. Thanks for joining us on this exploration. Did you learn something new about CRISPR? If you enjoyed this video, consider subscribing to SciSips for more in-depth dives into the fascinating world of science. Let us know in the comments what other science stuff you'd like to see.